this is going to just talk about DNA replication. So in the end of the last video, I talked about why DNA needs to be copied, which is basically what replication means. Like DNA copies the entire length of its molecule to make an exact copy, so that when one cell needs to divide into two cells for repair or regeneration or growing, that you have an exact copy of DNA in that new cell. All right, so how does DNA copy itself, right? So the term is going to be letter A, semi-conservative replication. So these are the steps of semi-conservative replication. The first image, we have a double-stranded DNA molecule. What happens in DNA replication is that the molecule will first break the hydrogen bonds between the two strands. Okay, so DNA is copied by breaking the H bonds and using each strand as a template. So the word template here is going to be used a lot in your book, and it's also going to be used on the test. It is what molecules, like enzymes, will read. It's going to be the sort of roadmap, right, or template of creating a new strand. What you see happening in step C is that there is a special enzyme called DNA polymerase, that you need to remember this name. DNA polymerase is an enzyme that will come in, read the template, right, which is in dark blue, and then add the complementary bases as it goes. So there's an enzyme, this DNA polymerase is pretty remarkable because it can create this new strand of DNA. And it does it simply by reading the old strand, or what we call the template strand, and then just adding on our complementary bases. So that it does this for both sides. And then when we're done, in letter D, when we have completed this replication process, we have two new molecules of DNA, right? Each of this dark blue, light blue, that's one molecule of DNA. It's a double-stranded molecule. Light blue, dark blue, that's another molecule of DNA. They are identical in terms of their sequence. It doesn't matter if they're light blue, dark blue, because in real life, these are just the same molecules anyway. These nucleotides, there's no difference. But what I want you guys to notice is the reason why we're using the term semi-conservative is because in these new DNA molecules, you'll notice that half of it, the dark blue, is conserved, right? We saved this half from the parent strand. And then the other strand is going to be completely newly made from scratch. So the word semi-conservative refers to the fact that your brand new DNA molecule will have half the strand conserved and half the strand completely new. So it's not completely conservative, it's semi-conservative. So just remember that's what semi-conservative replication means, right? You end up with a DNA molecules after replication using and having half of that strand, or half of the molecule, or one strand being old. Okay, so this is an image of what the DNA replication process looks like. The old, what we call the parent strand of DNA, is unzipped, so we take the hydrogen bonds apart, or break the hydrogen bonds, I should say. Then the new strands, the daughter strands, are being created by that DNA polymerase. There's a couple of more terms for you to know. So letter B, 1, 2, and 3. When DNA is copied, it actually gets copied at these places called the origin of replication. So it's not random. There are specific origins of replication where the machinery, and when I say machinery, I mean enzymes, will go and target, and they will open up the DNA two strands at those origins of replication. So what we've made is a bubble. And so this is called the replication bubble, so number 2. And the orange daughter strands, right? Remember, DNA polymerase is making that daughter strand. We can see that these two replication bubbles are going to work in both directions. So the arrows will show you that they're going to copy DNA in both directions. And each bubble has a fork, right? Just like a fork in the road. So this corner of the bubble, where replication is going to proceed in this direction, and the other fork, right, in the road here, the replication is going to go in this direction. So what we say is one replication has two forks, right? There's a fork on this side There's a fork on the other side. And what happens is, as the forks open up, the bubbles get bigger and bigger and bigger, and maybe you can imagine in your mind how these two bubbles can come together in the middle, right? When they meet in the middle, you're going to have those enzymes continuing to make those new orange lines, right, the new strand. And then we can see that we're going to have two new molecules of DNA made in that semi-conservative way, where the light blue is conserved and the orange is completely new. All right, so let's look at letter C, DNA polymerase, which is abbreviated DNA pol. It is an enzyme for making the DNA polymerase. It's an enzyme for making the DNA strands. And number 2 is important. It has proofreading ability. So when DNA polymerase makes a mistake, it recognizes the mistake or the mismatch, and it can fix the mistake. All right, so here's your question for you. A scientist inserts radioactively labeled DNA into a bacteria. When the bacteria reproduces, how much radioactivity will the DNA in the two daughter cells have? So this parent cell here has 100%. All of the DNA, both strands, have radioactivity on them. When that DNA gets copied and puts each one copy of one molecule in each of those new cells, how much radioactivity is left in those daughter strands of DNA? So if you answered 50%, that's correct, right? Because remember semi-conservative replication. If these light blue strands are completely radioactive, after you replicate that DNA molecule, you're going to have two new DNA molecules, but each one conserving radioactivity from just half the strand. So this molecule here would contain 50% of the radioactivity from the parent. Okay, good. So we're going to stop here and pick up genetic expression in the next lecture.